Hi, everyone, and welcome to the Faculty Friday Research Seminar Series. My name is Anne-Marie Parker, and I'm a Research Associate at the Joplin campus of KCU to Dr. Staudinger. I ask that everyone please stay muted for the presentation and save your questions for the end. I also just want to make everyone aware quickly that starting next semester, this research seminar series will be moving from 3 p.m. to 4 p.m. So the rest of the fall semester 2021, they're still going to be at 3 p.m. But starting then spring 2022, the time will change to 4 p.m. So if this is something that interests you maybe in presenting, or someone you know that you would maybe like to present, that's just something I want to make you aware of to note. So now, without further ado, Dr. Sharon Gordon will be introducing our speaker for the day.
Thank you, Anne-Marie, and thank you, Dr. Staudinger. And thank you, Dr. Baker, for being our presenter today. I'm very pleased to introduce Dr. Olga Baker. She earned her dental degree and her PhD from Central University of Venezuela, followed by conducting postdoctoral fellowships in both biochemistry and pharmacology at the University of Missouri. She's held academic positions at SUNY Buffalo and at the University of Utah prior to her present position as professor in the School of Medicine at the University of Missouri, where her research focuses on the inflammatory aspects of salivary dysfunction. She has 3 active NIH-funded projects and holds 4 patents related to salivary tissue regeneration. Among other awards and honors, she was named as Salivary Researcher of the Year by the International Association for Dental Research. She's an active leader in the American Association for Dental, Oral, and Craniofacial Research, serving as faculty advisor to the Student Research Group, treasurer on the board of directors, and she's the founder and the president of the Missouri section of the AADR, of which KCU has joined as a member this year. Her topic is very exciting to many of us who are interested in salivary function, as well as how it affects our patients. So I will now turn it over to Dr. Baker. Thank you, Dr. Baker.
Thank you, Sharon, and for the nice introduction and for giving me the opportunity to present our lab's work, my lab work here. And then, so you just heard, I am a researcher at Missouri, but before that, I was a clinician in my home country, Venezuela. And my work with head and neck irradiated patients and Sjogren's syndrome patients in the clinic is what led me to see the need for better hyposalivation treatments. So that's why the presentation today is novel approaches to promote salivary gland, or to promote function in damaged salivary glands. Let me see. Okay, so here we can see the 3 pairs of major salivary glands in mouse and human. I'm showing mice because this is the main model we use in the lab. And here we can see humans. In human and mice, we have 3 pairs of major salivary glands that produce 90% of the saliva we produce daily. And we also have minor salivary glands that contribute 10% of our saliva. And we produce saliva mostly when we're talking or when we're chewing. At night, we don't produce that much saliva. You know, some people think that because we drool when we're sleeping, it would be that a lot, but no, we mostly do produce saliva during the day. And in mice, different from humans, we can see this big salivary gland here. Can you see my pointer? Okay, so we can see the submandibular gland is the major contributor of saliva, and that's why submandibular gland is the main model we use in the lab. In human, the main contributor is the parotid gland, which is the biggest salivary gland we have. So in this little movie, which was developed by a dental student, actually, I work with a dental student, we show saliva is produced by activation of 7 transmembrane receptors that leads to the transepithelial gradient that increase sodium and chloride, produce a hypertonic saliva. And when the saliva goes through the ducts, there is reabsorption of sodium and chloride and secretion of potassium and bicarbonate. This is only showing for fluid. We also have other neurotransmitter, in this case is acetylcholine, but we also have adrenaline or norepinephrine that is important for protein secretion. So what happened when we lose our salivary glands? So when we lose the salivary gland function, of course, we lose saliva. The patients will have an altered sense of taste. Everything will taste funny. They have an increased susceptibility for oral infections. They cannot swallow. They cannot talk like I'm talking to you right now. And one very, very difficult thing for most of these patients is that they have trouble sleeping, because our saliva, lubricates all this area, and then they just feel like choking. So overall, these damaged salivary glands just basically lose, a patient without saliva have a very low quality of life. And there are no treatments. You probably prescribe to some of your patients like saliva substitutes, or say anything that tries to pilocarpine to try to promote saliva secretion, but they don't work, and that's a big problem. So in the lab, we work with two major problems. So the first problem I have here is Sjogren's syndrome, which is an autoimmune disease that affects the exocrine organs. And for this Sjogren's syndrome, we use specialized pro-resolving lipid mediators. Oops. And we also work with patients that have been irradiated for treatment of head and neck cancer. And then for these patients, we use, or we're trying to develop therapies using fibrin hydrogels and cell sheets. So here we can see a picture, actually is courtesy of Stefan Ruhl from SUNY Buffalo. We can see the oral cavity of a patient with Sjogren's syndrome. This is extreme. I just wanted to highlight the importance of the saliva here. The patient have extensive tooth decay. Not all the patients that have Sjogren's syndrome will present that severe clinical feature. In fact, some people like Venus Williams, she was diagnosed with Sjogren's syndrome in 2012 when she quit. She had to quit the U.S. Open, and then she has a healthy life. People can, there are things that people can do to maintain the dental health, but it still is a problem. Nobody knows what causes Sjogren's syndrome, and nobody knows how to cure it. The people believe, or the current research, which is from many years ago, is Sjogren's syndrome is caused by environmental causes, genetic, and but nobody knows exactly what is the cause of this disease. But what we know is that in Sjogren's syndrome, there is extensive lymphocytic infiltration of the salivary glands. Here's a picture that we took from a minor salivary gland from a Sjogren's syndrome patient. And we can see these blue dots. These are the lymphocytes. They are stained with a nuclear staining. And you can see that it's extensive. And then you can see on this side, you can see some ductal cells. And then if we see it bigger here, we can see adherent junction protein called ec-adherent and a tight junction protein called ZO1, which are polarized. Here we can see that they're losing polarity. Polarized means that the green should be inside, the red should be outside. And then, so this is not continuous. And then, so in a non-human, non-Sjogren's syndrome salivary glands, you can see a beautiful continuous tight junction, ZO1, which is in green, and a red ec-adherent. These are some ductal structures, so the tight junctions are intact. So most of the things that I will explain will have tight junctions on it, because tight junctions are important. Actually, let me go back here. If you see these, we can see here that the tight junctions are important for the transepithelial gradient. That is the main thing to produce the watery saliva. And that's why I highlighted here. And let me go back to the next one. So one day I was in a Sjogren's syndrome patient conference when I was a postdoc, and I started talking to some people that had Sjogren's syndrome, and I asked them how do you get by? And, what treatment, what sorts of treatments do you do? And many of them mentioned omega-3 fatty acids. So after I came back to the lab, I started looking links between omega-3 fatty acids and inflammation. So that's when I came to the work of Charlie Serhan, and then who works in resolution of inflammation. So resolution of inflammation is a new concept. Here we can see that when there is tissue injury or infection, there is a vascular response, and there are some chemoattractants here that leads to neutrophil extravasation. And in the past, people had the concept of inflammation as a passive event, meaning that once these prostaglandins and chemoattractants dilute, then the inflammation is gone. But nowadays, we know there are some lipid mediators called resolvins and lipoxin. And these lipid mediators are important for resolution of inflammation. Basically, one of the main functions of these lipid mediators is to promote the transformation from non-phlogistic monocytes to resolving macrophage, which are important for apoptosis of for phagocyting apoptotic neutrophils and leading to the formation of resolving exudates. And this also, so when they are working, the tissue will go back to homeostasis. When these resolvins or lipoxins are not working, there is chronic inflammation and that leads to organ fibrosis, autoimmunity, and all these problems. This is a highly simplified explanation. Basically, resolvins are important because it's an active process. We produce these lipid mediators, and these are important for resolving inflammation. And so what did we do in the lab? So once I knew, I contacted Charlie Serhan from Harvard University. He talked to me, he said, well, you have to try all these resolvins in your system. So I tried them all, and the one that worked was this resolving D1. Here I'm showing you the two main metabolomes from EPA and DHA. There is another one from arachidonic acid that I didn't bring, it's not in the pictures, but one of the most important resolvins are resolving E of the E series, resolvins of the D series, protectins, and maresins. So we work in the lab with most resolving D1. And this is the one that has shown effects. But people have worked in maresins, for instance, and they are important for tissue regeneration. These are for brain injury, neuroprotectin and protectin D1. RBE1 have been shown to be important for resolution of inflammation during periodontal disease. And besides these forms that you see here, there are aspirin triggered forms, meaning that when the patient takes aspirin, there are some other forms that are more chemically stable and last longer in the body. So if we see salivary glands, salivary cells, these are rat parotid gland parasitic cells. When they are plated on matrigel, they form nice spheres. And you can see here, if we would stain for ZO1 in green, would be here, adherent would be outside. So you can see a nice lumen formation. This is a healthy salivary organoid. And then when we, in the presence of inflammatory cytokines, if we add TNF-alpha to the culture, the cells are gonna go everywhere, they're gonna migrate, but they're gonna die, and they do not form that lumen. But when we treat these cells with TNF-alpha and add resolvins, these resolvins allow the ability to recover this lumen. That was a very exciting result. So we use this treatment also in primary cells. and found out that they are important because they block cytokine signaling in salivary glands when used in vitro. And so that was very exciting. And then I didn't want to bring all the experiments because it was gonna be like 2 hours. So I just, to simplify, I don't know why this one here. To simplify, we demonstrated that ATRVD1, the aspirin trigger form, when binds to salivary epithelium, activates these receptors called ALXFPR2, and in human is called GPR32, and activates a protein called GQ11, leading to this PI3 kinase, AKT. And these signals not only enhance cell polarity and cell migration, but they also block the signals caused by pro-inflammatory cytokines such as TNF alpha. So TNF alpha activates, I don't know why this thing moves, I'm not gonna touch it. So ATRVD1 blocks cell death by blocking caspase activation and enhance cell polarity and cell migration. Okay, now we can, but this was not enough because those are in vitro results. So we wanted to see whether this treatment was good in vivo. For these studies, we use a mouse model called NOD, non-obese diabetic. For some of you that are involved in research, this is a mouse that was generated a long time ago in Japan. It's caused by a spontaneous mutation of female NOD. They call non-obese and diabetic because they display diabetes. So when the mice are babies, when they are pre-disease, they have normal saliva secretion and their salivary glands are intact. At 16 weeks, we call the onset disease, they will decrease their saliva's production and their tissue will be inflamed. So what we did is we treated, oh my God, let me see here, sorry about this. We treated these mice with a vehicle, like ethanol, is where the resultants are dissolved. And we treated them with ATRVD1. And then we waited 2 months and then we studied them at advanced stage. So when they are older. And then so we collected saliva secretion, we performed histological analysis, confocal analysis, and cytokine profile. So you can see here, we in males, ATRVD1 treatment after disease onset was able to restore saliva secretion. So we can see here that males, this is the control, this is vehicle, and this is ATRVD1. And in females also, we can see that significantly, increased saliva secretion, what I'm talking about. So it increased saliva, restore the ability of the mice to produce saliva. So ATRVD1 was able to restore saliva secretion in these mice. So that was good. And then we say, okay, let's look at the tissues. But when we look at the tissues, we didn't see anything. We can see here a salivary gland. This is a submandibular gland, and it's attached to sublingual gland here. And we can see these darker spots. These are lymphocytic infiltration. And here we can see lymphocytic infiltration. In males, we also see some lymphocytic infiltration here. So, but the ATRVD1 treatment did not do anything. So that wasn't exciting. So we decided to study what happened if we study specific cell types within these infiltrates to see whether they are diminished. So we studied Th17 cell infiltration. Why Th17? Because previous studies had shown that this is the main cell population associated with Sjogren's syndrome. So we study the cell number. We quantify the cell number by using specific markers for Th17 cell. And we found that the number of cells that were positive for Th17 were decreased. So that showed that this particular cytokine was important and it was decreased. And previous studies have shown that this Th17 also is important because mouse models when you block Th17 in the cells, it preserves the tight junctions. So preserving saliva secretion, then we are diminishing the Th17 cell infiltration, and we are maintaining the tight junctions in the animals. And then we also study the particular cytokines. We can see a significant decrease of interleukin-17 in the female mice. I'm not gonna go over to the other ones. I'm just gonna concentrate on the Th17. So in summary, we saw that IL-17 is an inflammatory cytokine that is associated with Sjogren's syndrome and activates inflammatory pathways triggered by NF-kappa B. And resolving is able to activate PI3 kinase, AKT, and by this it blocks Th17 signaling. To establish the importance of the resolving receptor, we use a mouse that doesn't have the receptor for resolving. So we use a mouse model from the UK, from Mauro Perretti, from Queen Mary of London. And so he sent us the mice, and we study what happens when you don't have the resolving receptor. So here is the mouse studies. We injected this mice with LPS, which is like imitating an infection process. And the mice that didn't have the receptor had exacerbated, had decreased saliva production, have alteration of tight junctions, have an increased cell death, and they had neuronal damage in their salivary glands. So all of these indicated that the receptor, if you don't have the receptor, you will have exacerbated responses to LPS. Moreover, when you let those animals grow, particularly the female, have a decreased saliva flow rates, and they have an increased number of B cell markers, which are important for Sjogren's syndrome. And then so we also, other studies that we've done, we have demonstrated all the machinery to produce ATRBD1 and RBD1 are expressed and functional in salivary glands. So it's a complicated study, but just to make the long story short, these are the pathways from converting DHA into RBD1. And one important enzyme in this process is 5-lipoxygenase activating protein called FLAP, which activates 5-LOX to produce resolvins. So we study all these enzymes to see whether the resolving production in Sjogren's syndrome was altered. And then we found out that this FLAP protein is significantly diminished in salivary glands with Sjogren's syndrome. Okay, so that, so in summary for this part, for the resolving project, we can say that ATRBD1 treatment diminishes Sjogren's syndrome like features in Sjogren's syndrome mice. The receptor for ATRBD1, ALXFPR2, regulates salivary gland innate. This is with the LPS, and adaptive with the aging mice. Human minor salivary glands with Sjogren's syndrome have a reduced FLAP gene and protein expression, and FLAP is the protein that regulates RBD1 production. So in summary, we still are working on this mouse model. We now, for the future, we're now using mice that have, we call it humanized mouse. It's basically a mouse that is a knocking mouse. It has the human receptor inside them, and then we're gonna see whether the human receptor works as well, as good as the mouse receptor does. And that is in preparation for more translational studies. And we are also using human tissues to see in three-dimensional cultures to see what are the effects of these resolvings in blocking inflammatory cytokines in humans. So those are our future studies. And then I'm going to jump now to the radiation therapy effects. So again, we can see the damaging effects of radiation treatment. People with head and neck cancer have a lot of problems. You know, they have dysphagia, they have fibrosis, they don't produce saliva. So it's very also very debilitating for these patients. So here we can see some mandibular gland from a non-irradiated patient. You can see the pink structure. So we can see here just the lobules of the salivary glands. This is part of a paper, that's why it has the red square here. But so just to show you in general how a salivary gland would look, 6 months after the patient have been irradiated, we can see a lot of adipose tissue and some collagen production around the lobules. And here is 6 years post-radiation. And we can see the extensive blue. This is a Mason tritron blue stain. And we can see here that this patient is probably not producing any saliva at all. And then we need to replace that tissue. And that's the goal of this project, to replace as much as we can of this damaged tissue. So the studies started when I was using these parasitic cells grown on matrigel. And again, the green is the Z01 and the red is occluding here. So these are rat parotid gland parasitans, but we also use mouse primary salivary glands. And the glands, they can make like a mini gland. So that is cool. But there was an RFA. I don't know if you guys are familiar with RFAs, but NIH was looking for people that work with bioengineers. They say, we are calling for application for people that work in salivary field to work with bioengineers. And I was in Buffalo back then. So I contacted Stelios Andreadis, who was a bioengineer working in blood vessels using fibrin hydrogels. And so I joined forces with Stelios and we got an R01 from this RFA. And then actually we just got renewed. So we got 3.8 million in this grant. So that's really exciting. And then I'm gonna tell you the story. So when these cells, you see them here, they are nice and round. They're growing on something called matrigel that comes from mouse tumor. But this substance, matrigel, or laminin 111 actually, is mostly laminin 111, is a protein that is produced during embryonic stage. We cannot put that laminin 111 in patients because that would cause tumor. It's tumorigenic. We cannot produce a pure laminin 1 because it varies from batch to batch. So I came up with this idea. What about if we take pieces of this laminin 1 and put it into the hydrogels? That's when I went to Stelios and say, hey, is this possible? So let me just go back to the next. So we try different pieces. This is the protein, the laminin 111. And then we try different pieces. And these two pieces, when working together, we call A99 peptides and YIGSR. These are two peptides that when we put them together, actually they enhance salivary gland organization and tissue regeneration. So basically we use fibrinogen. We chemically link these peptides, A99 and YIGSR, to the fibrinogen. So we form fibrinogen linked to these peptides. And in another side, we use daylight 680. We conjugate it to fibrinogen. And why we do this? We do this to track the gel in the animal. And you see very cool pictures. So here's how it looks like. I would say like in a, I guess, microscopically, or would look like that. It's a gel. So we use a mouse salivary gland. We made a hole and we add the scaffold here. So this scaffold will activate the epithelial cells and will lead to cell adhesion, migration, and proliferation. So when the cells are grown on fibrin hydrogel alone, so fibrin hydrogel again is fibrinogen with thrombin, it's just a hydrogel. They grow flat. When you put those peptides, and we try different combinations, it's not like this happened from one day to the next, but we try other peptides. But when this particular combination of peptides are bound to the fibrinogen, we form the sphere. And that was awesome. We were like, wow, this is like really cool. So we demonstrated here that these two peptides were important for cell migration and organization. Then we created a wounded mouse model, as I mentioned to you. So basically, we open the mouse, we make this punch, we inject the hydrogel here, and then we put it back, we sew it back. Then we take it out and study after some time in treatment. We got I put the picture of the lab because we got a prize. We got the GS award in for this, I think was a 2017 paper. So anyway, then we can track the hydrogel. So you see in day one, this, remember when I say daylight 680, this is the daylight 680. So we can use the dye to track where the hydrogel goes. So these small in day 3 and really small in day 8. That means that the hydrogel shrinks over time, which is what we want, delivers the peptides and is gone over time. So that was good. And then when we take the glands out, I want you to point your attention, call your attention in this one. It's called sham. Sham is a gland that you take out and put it back in, you do nothing. And then this one is the submandibular gland, and this is the sublingual gland. When you make the hole, it's gonna look like this, with no scaffold. No scaffold at day 3, you can see this big hole still, and at day 8, you see extensive fibrosis. If you use a fibrin hydrogel alone, the pieces are gonna stick together, but at the day 8, they are messed up. There is no organization. When you add the peptides at day 3, the pieces come together, and day 8, it almost looks like the sham control. That was amazing. That was really, this is a really good result. And that's the one that got the award. So this showed that the laminin 1 peptide promote tissue regeneration in this wounded mouse model. Not only that, but also promoted saliva secretion as compared to control and the fibrin hydrogel alone. So those were really exciting results. And however, and then there are more results here. They promoted cell differentiation, cell polarity, and so forth. But this punch is not what we see in the clinic. In the clinic, what we see is head and neck irradiated patients. So we created a mouse model. When I say we created, it's not that we invented it. Many people have done this radiation model, but you have to adapt it according to your machine and according to the mouse environment. So it's that we have to set it up. So we took a mouse, we created a shield that goes in the glands, so we only get head and neck radiation. And then after 3 days, we inject with the laminin 1 peptides. Sorry. Sorry, then we do our experiment at day 8 and at day 30. So here we can see the mouse that is being irradiated. Then we can see here the hydrogel that we injected via transdermal injection. And we can see here that the saliva flow in the mice, these are the untreated mice, and these are the treated mice. The treated mice maintain their saliva flow rates. So that was really exciting. And we just published this actually a few weeks ago in the Journal of Frontiers. So that's an excellent, that's a really good result. It's very translational And then I wanted to show you the results. If you see a non-irradiated gland, I'm calling your attention to this day 8. You see a nice sublingual and submandibular gland. We can see it here again at day 8. This is with H&E, and this is with Mason trichrome, which is to stain for, the blue is the fibrosis. Then at day 30, and then so all of these are non-irradiated. So if you see across the tissues, you're gonna see they look normal. They look the sublingual and submandibular, They're very similar to each other. When you radiate the gland here, this is a day 8, a little bit of fibrosis here. Day 30, look how the gland has lost their morphology, and it has a lot of fibrotic tissue here. And you can see that this shape is no longer a shape. When we treat the mice with laminin-1 peptides, we can see the beautiful submandibular and sublingual. And it actually the gland almost stays the same. You know, it still has some fibrosis, and we still need to modify this scaffold. We need to make it better. We are trying to use some growth factors and perfection, make it better for more preclinical studies. So in summary for these hydrogels are amazing. And that's the one that we just got the grant review for these studies. So that's really exciting. So another part of this that I wanted to show you in the non-irradiated, this looks like very beautiful. This looks like a heart actually here. But the red is E-cadherin, it's a basolateral protein. The green is ZO1, it's an apical protein. So this tissue is intact. And this also is intact. This is sodium potassium ATPase, it's a basolateral protein, and TMEN16 is an apical protein showing salivary gland differentiation. So as you can see here, at day 8, you lose the polarity and differentiation, which is even greater. These are the irradiated areas. We lost all our differentiation marker here. But this will shrink when we treat the animals with the laminin-1 peptide transdermally. So we actually quantified this, and we found that the wound, this damage will shrink over time. And we now did 60 days, which is not here, which is even more drastic, better, even better. And then regarding saliva secretion, we can see here that if you irradiate the mouse, so this is a non-irradiated glands. This is when you irradiate it with 15 gray. I think this is, yes, this is 15 gray. You see the saliva flow rate decreases, still decreases. And at 30 days, we can see that this saliva secretion is almost non-significant with the non-irradiated saliva flow, with non-irradiated animals. So in summary, this indicated that the treatment with laminin-1 peptide fibrin hydrogels restore saliva secretion after day 30 in treatment. And so in summary for this part, we show that laminin-1 peptides, A99 and YIGSR conjugated to fibrin hydrogel promote formation of acinar-like structures in vitro. These peptides improve salivary gland regeneration. And this is an old conclusion because we already tested in the irradiated mice. So, but we are improving, current studies are improving these scaffolds. And then the last technology that I wanted to show you today is called the cell sheets. So basically we have a mouse, we take the salivary glands, we mince and we create like a homogenate of the tissue, and we plate it in something called thermoresponsive plates. So these thermoresponsive plates are very interesting because normally when you have the cells on a petri dish and you use trypsin, you lose these green proteins called extracellular matrix proteins. These thermoresponsive plates have the ability, when you decrease the temperature, they have a polymer that will hydrate and will release this monolayer. And in turn, this will maintain these extracellular matrix proteins that are important for tissue regeneration. So we can see here how the monolayer, the salivary gland monolayer, detach over time. And here is an H&E structure of the cell sheet. It looks huge here. It's a very interesting structure. So we wanted to try to see if those cell sheets are important for salivary gland regeneration. And again, we use the wounded mouse model. And this is how the cell sheet looks. And this is incredible. Once you add the cell sheets into the mouse, immediately attaches because it has tons of ECM, extracellular matrix protein. And then we close out the mouse. So we have published 2 papers on the cell sheets, one in 2019, one in 2020. And then here, the untreated mice, we can see extensive damage and fibrosis. If we tried it with one single cell, they have some regeneration. But if you tried it when we treat it with double cell sheets, the damage is actually less. And this is the sham control. So the double cell sheet treated mice are even better than the sham controls. And when we study saliva secretion, we can see the double cell sheet is almost close to the sham regarding the saliva flow rate. Let me see here. So these double cell sheets promote salivary gland tissue regeneration at a faster rate than the single cell sheets. These double cell sheets could be used as a strong in vitro model because right now we don't have in the salivary field a good in vitro model to study salivary gland structure and organization. And right now we are characterizing the mechanism how these cell sheets improve regeneration. The problem with the cell sheets are that salivary acinar cells are composed by acinar and ductal structures, and they are mixed together in the cell sheet. So we need to characterize how these cells change over time and how to use it better. What cell types are in there, and do we have more progenitor cells? Do we have stem cells in the cell sheet? What other things are there? So we're still characterizing these cell sheets. We have other things that I didn't talk to you today. We are doing collaboration with the Forsyth Institute. We are working with lipidomics in oral tissues to study which other resolvins are there in saliva and salivary gland from human subjects. We're also doing mathematical model to study how these resolvins can be used systemically, especially for the Sjogren's syndrome patients. And the third project is a workable mucin. So my lab, actually, we just got a grant. It's called the Triumph Grant. It's an internal grant. But back when I was in Venezuela, I used to make mucins for head and neck cancer patients. So I retook that route, actually had a company in Utah. And then so we are isolating mucins not only from animals, but also from plants. So there are mucin-like proteins. And then we're gonna start clinical trials soon. So those are the novel things I have in the lab that are currently the work. And so this is my current lab. This is the Utah lab. It's Buffalo lab. This is Utah lab. These are my patients, Sjogren's syndrome patients. Dr. Brian Trump, who's a collaborator. Oops. Let me see. How do I go back? Okay, here. And then I thank to all the support of NIDCR and of course the startup fund from the University of Missouri Columbia. I'm happy to take any question. Thank you again for inviting me, and that's it.
Thank you, Dr. Baker. That was fantastic. I know that Dr. Staudinger is going to ask if people have questions, and I do happen to have questions, but I will yield to others who may have questions first.
Yes.
Go ahead, Dr. Johnston.
Oh, he has mute. He's mute. You're muted.
Sorry. Dr. Baker, thank you very much. I really enjoyed your presentation, although I must admit, I got lost in a moderate amount of it. But from a clinician's perspective, I wanted to ask you some corollary. You know, you have studied, from a Sjogren's perspective, we teach that patients clinically will come in with dry mouth and dental caries and dry eyes as well. And I don't know this, you did show the picture of the mouse and the anatomy. Has there been much research on tears? And I say that based upon, that's a common symptomatology as from the eye perspective as is the mouth perspective. So can you, because you had implied that I think it's the flap gene that does help produce the protein, generate this. So what can you share your knowledge about studies on tears and correlate that with saliva?
Yes. So actually, I am working now with lacrimal glands. So because that is important actually for the renewal, although my grant will end in 2023, I am now getting ready to submit for the next round. And then for the next round, we have collected the lacrimal glands from the mice, but the weird thing is that they're totally different from the, let me show you here. So let me share the screen here. Let me see if I can share the screen. So we saw here that female have more lymphocytic infiltration than the male. That's similar to the humans, right? Like affect mostly women, like 9-to-1 women. But in male, the lacrimal gland, so it's puzzling. The lacrimal gland is almost, is a lot of lymphocytic infiltration, and the females are almost normal. So it's the opposite. They're showing me the opposite or the opposite clinical features as what I see in the salivary glands. So I don't know what to make of it, but I'm studying it. And they are related because they both exocrine tissues, they both produce fluid, they both associated with, muscarinic receptor signaling. So I am committed to study the lacrimal gland. So, and then we know in the eye, not only the lacrimal glands, but also the meibomian glands, like oil producing glands. There are other things. So I have Clarissa Petris, who works here at Missouri, that she's an ophthalmologist. So I always want to try to have clinicians in the grants, because they know more than me, about this. But I take your question very seriously, because yes, we need to know how they're both correlated. And actually, it would be nice if I could get tear samples and human lacrimal glands. I see a question from you. You're welcome. Irina?
Dr. Kennedy.
Thank you so much. This presentation was amazing. In your work with the hydrogels, I noticed that, before and after the treatment, you were looking at the quantity of saliva produced, but did you also look at the quality and function, meaning
we did.
There's certain like biological components, and were you still able to mount a buffering response using like the proteins that you have in saliva as well?
Yes. So the protein patterns is actually, I think we also published the protein patterns, and it was more not only the saliva flow rate, but also the protein composition, like the amount of different type of things like MOG10 or MOG1. We have done that before using SDS page. It is hard to quantify the really high molecular weight MGN1 and 2. That's hard because you have to do an agarose gel. We haven't done that, but we saw the general protein pattern is recovered when we treat the mice.
Thank you.
Well, Sharon?
Thank you. So in your radiation injury model, and when the mice were treated with the lamina 1 peptide, I was just curious about if you could give more details on the treatment protocol. So was it like pre-radiation injury or post? How many doses, and what was the delivery method?
Right. That's a question that everybody asks.
Because I know you're going towards treatment, you know,
Yes,
in humans.
yes.
And so I was just curious where you were with that.
Yes. So let me just get the here. Let me see if I can share this screen. So you have a mouse. We anesthetized. For this study, we did anesthetize, no longer anesthetizing. We now have some mouse restraint. We use a lead shield, which is this gray color that we put there, and it has an opening. Okay, so the day one, they receive a single dose of radiation, it's 15 gray. I know that the patient in the clinic is not like that. They have fractionated doses, but this is just for developing the model. Future study will do fractionated doses, but here we just did a single dose, is 15 gray, which we knew it caused massive saliva secretion defects and also destroys the salivary gland. So that's why we use the maximum just to create a model. Just say, we're gonna destroy the gland and try to cure it. Then so we did that day one. Day three, get the mice again, and then injected via transdermal injection using a very, very tiny needle. This is not perfect, but you see the red color here. This is where the biggest amount of the hydrogel is there. But then it leaves this tissue alone. So it's only gonna cure a piece of it. It's gonna only affect just a little bit. So we got a collaboration, actually, yesterday I had a meeting that we're gonna do ultrasound guided delivery of these hydrogels. The areas, when they get farther from this hydrogel area, they're not as good as this part. These parts are the best. But yeah, we need to improve the delivery method. So then after 3 days, we did a single treatment dose, and then we waited 15 days and 30 days. So we only did one treatment. So one radiation treatment, one the hydrogel delivery treatment. So.
And it's direct delivery into the salivary gland.
Yes, it's a needle that goes transdermally into the gland.
Okay, awesome. Thank you.
You're welcome. Any other questions? Okay, well, thank you so much for inviting me and giving me the opportunity. I'm open for collaboration and for tissues. You know, if you guys collect minor salivary glands, I'm always needing those.
No, that's really good to know, because we'll definitely want to have more conversation with you about that, especially when Dr. Abbott gets here. So that would be wonderful.
That would be awesome if we can get like minors, because we're so close.
Right.
You know, we can enhance collaboration, get some tissues published together. I think we can be a big force. And
yeah, fantastic.
Anybody wants to visit here in Columbia, you're invited.
Thank you. Well, I'm anticipating you might host when we start to have the in-person sessions for the Missouri section, right?
Yes, exactly.
Yeah, and we can always host too at some point, and
yes,
and our other partners as well. But yeah, this was so fantastic. I'm so pleased you accepted the invitation, and you're doing wonderful work of great interest to all of us.
Anytime. And you're welcome to all those nice notes from everyone. Thank you so much.